Hi everyone. This summer we investigated climate change impacts on farmed and wild oysters. Eastern oysters are experiencing instances of sudden unusual mortality syndrome, or SUMS, especially in the summer months. We want to study how environmental conditions in the summer might be affecting gene expression. Our primary goal is to identify any differences in gene expression between oysters at the Duke Aqua Farm and oysters at CMAS, which is NC State's marine lab. Our secondary goal is to analyze how patterns in gene expression might be correlated with environmental variables to help explain how oysters are responding to climate change and if these findings can be used to reduce mortality. We created an RNA sequencing pipeline, which we ran using 3 reference genomes at 2 farms. This pipeline ran differential expression analysis, grouped genes that behave similarly, then linked genes to environmental variables and their biological functions.
And now we move on to visualize some of the results from the bioinformatics pipeline. So first, we want to look at how gene expressions differed by sites and months. With figure 2, a pairwise volcano plot for August versus June at CMAS Farm that identified blue dots as the insignificant gene expressions and red dots as these genes with significant gene expression. And through the plot, we can identify a substantial amount of red dots on either sides, with a positive side of the x-axis representing genes upregulated in August. In figure 3, which separates dots by location and month, we can see how blue shapes are more clustered together, while the red shapes are more scattered. And these results can indicate potential seasonal effects on oyster gene expressions. Then we want to move on to observe how there seems to be a correlation to exist between the environmental variables. And figure 4 is a heat map of a Spearman's correlation, with the overall trend pointing towards a moderate positive correlation between pH and salinity and pH and dissolved oxygen. And there seems to also be a moderate negative correlation between temperature and dissolved oxygen.
Awesome. We then ran a weighted gene co-expression network analysis, which basically clubs genes into different groups called module eigengenes, and they are then tested against different environmental factors like salinity, dissolved oxygen, and seen how correlated these groups are to those factors. So one of our most significant results was ME Black being extremely correlated with salinity, which you can see has a very, very low p-value. And then to further test that theory, we then ran linear regression of ME Black against salinity, and you can see that there's a negative correlation to it, which means that every time salinity increases, ME Black, the genes expressed in ME Black decreases, and vice versa. We also then pulled out the individual genes from ME Black and then ran enrichment and pulled out the exact genes and ran that against salinity, and we were able to see very similar values in a few of those genes, which means that these results can be seen on a very individual level.
We also ran our pipeline on 2 other reference genomes for eastern oysters. First, we compared the alignment rates of our raw reads to each genome. The box plot suggests that the alignment rates to each of the 3 genomes are pretty similar, with the 2017 Rutgers University genome having a slightly higher alignment rate. Then, we performed a Kruskal-Wallis test on the alignment data and found that the 2017 Rutgers genome had significantly different alignment rates compared to the other 2 genomes.
And lastly, we ran a gene functional enrichment analysis on all 3 reference genomes with a list of go terms such as ATP hydrolysis activity and protein refolding that we observed to be similar between these, which could indicate how these results could have a reproducible trend and how oysters may be experiencing cellular stress with a need to maintain protein homeostasis.
So, what did we find from these results? One of our major findings was that location and month and environmental factors truly do affect these genes and how they're expressed. We used WGCNA to correlate them against different environmental factors like dissolved oxygen, salinity, month, and conductivity. One of our major findings was that the black module showed a significant association with salinity with a very, very low p-value and a high R squared. And these patterns can help us understand that there can be early stress within these oysters and that rapid environmental change can truly affect these oysters.
Our work this summer will provide the Wong Lab with a pipeline to perform similar analysis on tissue sample reads and environmental data from 2025 to see if the patterns we have observed from 2024 are continued over time. Overall, we hope that our findings with the impact of environmental factors on gene expression will help future researchers understand why SUMS might be happening and how to reduce it. Thank you.